Hello, I'm Dr. Melissa Nelson, a postdoc working with Dr. Rhonda Souza and Stuart Spechler in the Center for Esophageal Diseases at Baylor University Medical Center and in the Center for Esophageal Research at the Baylor Scott and White Research Institute. The editors of Neurogastroenterology and Motility have asked us to discuss a report entitled Lower Esophageal Sphincter Muscle of Patients with Achalasia Exhibits Profound Mast Cell Degranulation. Achalasia is a serious esophageal motility disorder that is characterized by loss of peristalsis in the body of the esophagus and by failure of the lower esophageal sphincter to relax with swallowing. Patients with achalasia frequently experience terrible difficulty with swallowing, regurgitation, and aspiration of material that is retained in the esophagus, and chest pain. Long-term complications can include malnutrition, aspiration pneumonia, and even esophageal cancer. The cause of this devastating esophageal disease is not known. 2 years ago, our group at Baylor University Medical Center was the first to propose the novel hypothesis that there is an allergy-mediated form of achalasia. When we first proposed this hypothesis, it was based on studies that had found dense accumulations of eosinophils in the esophageal muscle of some achalasia patients, and it was based on reports of patients who had eosinophilic esophagitis, EOE, that was associated with achalasia, but whose achalasia disappeared when they were treated for EOE. Activated eosinophils release a number of products that can affect esophageal smooth muscle and the myenteric neurons in the esophageal wall that innervate the muscle. If eosinophils were to accumulate in esophageal muscle, their degranulation products conceivably could cause achalasia-like motor abnormalities. Since EOE is regarded as a disorder in which food antigens induce a type 2 allergic immune response, we propose that there might be a muscle-predominant form of EOE allergy that manifests as achalasia. Esophageal muscle is not accessible during routine endoscopy, and so a muscle-predominant form of EOE that causes achalasia might go unrecognized. That was the rationale for this study, in which we took biopsy of LES muscle from patients with achalasia or EGJ outflow obstruction who are undergoing Heller myotomy for treatment. When we started this study a few years ago, we were focused on the role of eosinophils, but recent evidence suggests that mast cells, the other key effector cells of allergy, also play an important role in the pathogenesis of EOE signs and symptoms. Like activated eosinophils, activated mast cells also release numerous myoactive, neuroactive, pro-inflammatory, and toxic products. So all the arguments we propose for how eosinophils in esophageal muscle might cause achalasia also pertain to mast cells. Eosinophils are usually very obvious in tissue specimens stained routinely with hematoxylin and eosin. But to see mast cells, you have to do special stains like immunohistochemistry for mast cell tryptase. We obtained LES muscle biopsies from 10 patients with achalasia and 3 patients with EGJ outflow obstruction who were undergoing Heller myotomy. For controls, we used LES muscle from the esophagus of 7 heart-beating deceased organ donors who had no history of esophageal disease. We stained the tissue with hematoxylin and eosin, and we performed immunostaining for mast cells. Remember, we were looking primarily for eosinophils, but we found that eosinophils, in fact, were rare in the LES muscle of both patients and controls. In contrast, mast cells were plentiful in LES muscle, but we found a huge difference between patients and controls in the mast cell degranulation status. 12 of our 13 patients exhibited profound mast cell degranulation in LES muscle, with copious amounts of tryptase-positive granules in the paramecium and around the nerves in the myenteric plexus. In contrast, we found evidence of only very mild mast cell degranulation in 2 control subjects, while the other 5 had no mast cell degranulation at all. We also evaluated our LES muscle samples by qPCR for the expression of genes involved in calcium handling and smooth muscle contraction, and we found that patients fell into one of 2 distinct gene patterns that we called mototypes. We found that these 2 genetic mototype patterns could distinguish among the 3 manometric phenotypes of achalasia. Our most striking finding was the profound mast cell degranulation we observed in LES muscle in 12 of our 13 patients. Mast cells are well known to play a pivotal role in allergic diseases in which IgE triggers their degranulation. This finding of profound mast cell degranulation in achalasia LES muscle provides support for our hypothesis that achalasia can be an allergy-mediated disease. However, these findings are not proof of underlying allergy because mast cell degranulation can be triggered by physical stresses and by a wide variety of non-allergic stimuli. So it's possible that the mast cell degranulation we observed in achalasia LES muscle was not the cause of myenteric plexus inflammation, but merely an effect of myenteric inflammation triggered by some other underlying condition. Although our findings are not definitive, they do provide considerable further support for our hypothesis that achalasia can be allergy-mediated disease in some cases, and they suggest that when patients acquire the mast cell degranulating form of achalasia, their underlying genetic motor type might contribute to the resulting manometric phenotype. Thank you.